Okay, well, it's a pleasure to be here, and I really appreciate Felicia's presentation before mine because it gives a lot of introduction to what we're going to be talking about in this presentation, because ViaCyte is also a company that is working on a stem cell-derived treatment for type 1 diabetes. We are looking for a functional cure, and we are in the clinic, so I'm going to talk a bit more in this presentation. I'm not going to give you the kind of background you just heard, but talk more about what we're learning from being in the clinic with these types of programs. So the technology we're pursuing is a stem cell-derived islet replacement therapy. We start with an embryonic stem cell, and I'll talk more about that. And we also have a proprietary cell delivery technology that we are using. We have two products at the clinical stage. The first one we call PEC-Direct. It is a first-generation product that is designed to be used with immunosuppression. So it's targeting the high-risk patient population, about 10% of the type 1 diabetic patients out there. We also have a second product that we call PEC-Encap. This utilizes an encapsulation technology that is designed to protect the cells from the immune system. We actually have clinical data showing that it in fact will do that. And we consider it a second generation because it is a more challenging product that we're learning from and improving in the clinic. I'll talk more about that. The competitive advantage that we have, we've been working in this field for about 10 years. We collaborate with CIRM and JDRF. We just announced last week a $20 million grant from CIRM for the PEC-Direct program. So we have a lot of technology and know-how leadership after over a decade of working here. We have a very dominating intellectual property portfolio. We have over 500 issued patents worldwide, including over 100 issued patents in the U.S., and still 800 pending applications worldwide. And we were the first company to move into the clinic with a product for diabetes or with a combination product, live cell product like this. The key messages continuing, we have a potential for accelerated development timelines, both because of the clear, unequivocal efficacy endpoints that we deal with type 1 diabetes, and also our expectation of having breakthrough therapy designation next year. These represent major commercial opportunities. Obviously, a product that can treat all type 1 diabetics as well as insulin-dependent type 2 is a huge opportunity. But even the targeted product for the high-risk patient population is a significant commercial opportunity. And we have a number of value-driving milestones, all clinical-based, coming up over the next 6 to 18 months. So in the last talk, there was a discussion of cadaver islet transplant, so I won't spend too much time on this, but there is this proof of concept that you can cure type 1 diabetes with islet therapy, cell therapy. So this comes from using cadaver islets. It often takes more than a single pancreas to get enough islets to treat a patient, but it is a very effective cure. In fact, a paper came out recently that showed that some of the patients who got this 18 years ago are still cured, insulin independent. And most of the patients that receive it, the majority these days go 5 years or longer insulin independent. So it's very effective, but as you already heard, it has a very lot of limitations associated with it. Supply is the big one. Getting a high-quality donor tissue is a challenge. It does require immunosuppressive drugs. It's very costly. So the technology we're developing at ViaCyte are designed to eliminate all of these limitations. With PEC-Direct, we would have an unlimited supply of cells, high quality, at a more reasonable cost, but still need immunosuppression. And then the encapsulation, the PEC-Encap product, is designed to move away from the immune suppression. So this slide very quickly talks about the various parts of the technology, the legs of the stool, if you will. One is we have a renewable cell source, a human embryonic stem cell line that we built a number of years ago. We have this banked in master and working cell banks. This line has been carefully studied. It's been evaluated and tested thoroughly. We've had derivatives from this line in 4 GLP safety studies, long-term studies. and it's been reviewed by regulatory agencies in the U.S., in Canada, and now Europe. And so it's a very well-characterized line as our starting material. But the next part is differentiation up to what we're going to do in plant. This represents about 7 or 8 years of work to go from that embryonic stem cell, drive it through a series of different cell steps up to a pancreatic progenitor cell. This is a cell line that is essentially destined to go on to form all of the cells of the islet after it's been implanted. So it takes about 2 to 3 months after implantation, and it matures and forms all of the cells of the islet, including the important beta cells. The delivery devices I'll talk more about in a minute, but that's another leg of the stool that allows us to deliver these cells to the patients effectively. So we're using 2 devices. The 2 products, PEC-Direct and PEC-Cap, use exactly the same cellular material, but they differ in the way they're delivered. So the PEC-Direct has an open device. It has ports that we engineer into it, a very discrete number of ports that we engineer into the surface of the device, and that allows direct vascularization, very much like those cadaver islets. And that direct vascularization gives a very robust implant, but it does require that immune suppression. The PEC-Cap has a semi-permeable membrane that is designed to protect the cells, so basically insulin and glucose and oxygen can move freely across that membrane, but blood vessels can't get in, cells can't get in, and that way it protects the cells from the immune system. However, it does require this vascularization, as shown in this slide, to form on the surface of that device, so it's much more susceptible to foreign body response, and we'll talk more about that in a minute. So both PEC-Cap and PEC-Direct are effective in animal models. We've looked at a wide variety of different species in rodent models, knockout animals. It will control blood glucose at or near the human set point. In fact, when we put this into an animal, we humanize that animal. It will regulate the blood glucose level to the human level for the life of that animal. It will appropriately release the insulin in response to blood glucose. We've never seen any evidence of hypoglycemia. One thing we learned early on is that different models, it will respond differently, and it seems to be driven by the aggressiveness of foreign body response. There's many models out there that you get a relatively mild foreign body response. It's very effective in those models, but we've also seen models where you get a very aggressive foreign body response, and it's much less active when we're talking about the PEC-Cap product. So we needed early on, we realized we needed to get to humans to really understand what was going to be required to make this an effective product. So we decided to move forward with PEC-Cap into the clinic a couple years ago. At the same time, we were working on PEC-Direct because we knew if we saw that aggressive foreign body response that we had more work to do. So there we go. So we initiated an open-label study with PEC-Cap in patients with established type 1 diabetes to develop the methods and procedures to minimize the trauma, and also, importantly, to assess that foreign body response. Simultaneously, we prepared to develop PEC-Direct should we see a more aggressive response into humans. So just to give you a quick overview of what we learned when we went to the clinic with PEC-Cap, this is based on 19 patients that were studied. Some of the key findings was we showed that it was safe and well tolerated, no AEs related to the cells. The AEs we saw were related to the surgical procedure. The encaptor cell delivery device did exactly what it was designed to do. It protected against the adaptive immune system. We saw no evidence for any allergenic or autoimmune rejection or even sensitization. So we were very pleased that encaptor was doing what it was designed for. We did see the potential for longer-term cell survival, and you can see that in the first 2 panels there. Those are 12-week devices that have been pulled out and subject to histology, and we can see good cell survival, islet-like tissue. When we stain those, immune stain those, we can see that there's beta cells and alpha cells in the graft. We see vascularization. However, those were not the rule. Mostly what we saw was in that last panel there, which is a very active foreign body response, laying down a layer of cells on the surface of that membrane and therefore blocking that vascular network that we needed and leading to a loss of cells inside. This was more like what we saw in the more aggressive animal models. So we had our answer as to where the humans sat. So based on that, we pushed forward with PEC-Direct aggressively. The reason we're doing that is it's not affected by this foreign body response. This slide shows on the left-hand side there, you see an animal model with an aggressive foreign body response, very similar to what we saw in the humans. In that very same model, when you put PEC-Direct in there, where you have these ports that allow the direct vascularization, even though you're still getting the same foreign body response to the device, it has no impact on the cell graft. You see a very robust graft. You can see evidence of the vascularization inside that device. And so those cells do very well. This works just as effectively in the models where you have a mild foreign body response and it works very effectively when you have the very aggressive one. So based on that, we moved forward with PEC-Direct into the clinic earlier this year. We filed INDs in the US and Canada, CTA. Those were approved, and we have now initiated that trial. The goal is to do 6 patients at a subtherapeutic dose. We put in Sentinel, small devices that we can pull out periodically to see how that engraftment and maturation is going. We're doing that now. We pull them out at 4, 8, 12, 16 weeks. So we've got the first group of patients implanted and are starting to get that data back. It's still early, but I'll show you a little bit of what we're learning. The cohort 2 is expected to start fairly soon, as early as next month, and that's where we will give a fully therapeutic dose and look for efficacy. And that efficacy, the primary efficacy endpoint is C-peptide levels, which is a biomarker for endogenous insulin production. These patients are C-peptide negative on enrollment, so if we see C-peptide at significant levels, then the product is doing what it was designed to do. So this just shows a timeline for that study. I won't go through that timeline, but just point out a few of the key milestones. By the end of this year, early next year, we expect to have the histology data from the first groups of patients showing that we have achieved the engraftment that we expect with PEC-Direct and showing that those cells have matured appropriately. So that will be an important proof of concept. And then by the end of the year, we expect to have the first 10 to 12 patients that are dosed with the full efficacy dose, and we will measure efficacy 6 months later. So about middle of next year, we would have our early demonstration of efficacy. We'd use that to file for breakthrough or RMAT designation. And then definitive proof of efficacy for this trial would come when we have 6-month data on all 36 to 40 patients in cohort 2. We expect that around the end of next year or early 2019. Just to show you a little bit of data from the current trial, this is looking at 4-week Sentinel explants. So again, these are small cell-filled devices that we implant and then we pull back out and subject to histology. This is a low-powered view, but what it shows here, at 4 weeks, it's still too early to say too much about this, but we're seeing very good engraftment, very good integration into the tissue with these 4-week pulls. Looks much better than what we initially saw, for instance, with PEC and CAP. So we're really encouraged by this, but it's still early days, and we need to get out to the 12-week and longer data to really show that we're achieving what we set out to do. So PEC-Direct has a lot of advantages. It has the low technical and execution risk based on the proven efficacy of cadaver islet transplants with that direct vascularization. The results with PEC and CAP clinical development showing that we do get in vivo maturation when the cells are vascularized, and the extensive preclinical data showing that there's no impact of foreign body response on PEC-Direct. It has an accelerating cost-effective development program, and we brought in Navigant to do a commercial assessment of this, and they came back to our board and reported about a $1.6 billion forecast, revenue forecast for the U.S. alone, so it's a significant commercial opportunity. We are still, we're aggressively pursuing PEC-Direct, but we have certainly not taken our eye off of the treatment for all patients. We are continuing to optimize for the second generation product. We're continuing to work with PEC and CAP. We formed a collaboration with W.L. Gore. They are the world's leader in fluoropolymers and fluoropolymer implant technology. And the membrane that we have in our device, the encaptor device, is essentially Gore-Tex. And so they are the world experts in that, and we're working with them to make improvements on that device to bring PEC and CAP back into the clinic. The IND remains open, the study remains open, so we believe we can do that fairly rapidly. We are also working on gene editing. We have a project we call PEC-QT, where we're looking to take our very well-characterized stem cell line and edit it to make it less immunogenic, and then use that to develop a treatment that could be done in an open device, as we've talked about with PEC-Direct, but without the immune suppression. So this is the progression we see going from PEC-Direct, targeting early on the kidney transplant patients that are in need of a treatment, going to the hypoglycemic unaware patients. Is 10% of the patient population that have a high incidence of serious adverse events associated with the insulin therapy. And then moving with PEC and CAP and or PEC-QT to all patients that require insulin. So I'll stop there. The key messages are, as we said, we are a leader in this field. We have 2 products at the clinical stage. We're generating data that you cannot get from animal models. We have accelerated timelines, very significant commercial opportunities, and a series of really important value-driving milestones, clinical milestones coming up over the next 6 to 18 months.